Good afternoon, dear colleagues. It's my great pleasure to welcome you to the webinar organized by the European Reference Kidney Network. The topic of today is Mendelian and not Mendelian inheritance, and will be presented by Dr. Kalman Tori from Budapest, Hungary. My name is Elena Levchenko. I'm a pediatric nephrologist in Leuven, Belgium, and I will moderate this webinar for you today. As you know, during the webinar, you can write your questions during the whole presentation in the attendee panels, and we'll try to answer your questions at the end of the webinar. The webinar will be recorded and the slides will be published on the WorkNet website, and you will also have poll questions, so be attentive and try to answer them all. Let me introduce the speaker of today. Dr. Kalman Tori graduated from Semmelweis University in Budapest as MD. During his postdoc training, he studied the genetics of nephronophthisis and steroid-resistant nephrotic syndrome in Paris in a very known laboratory led by Corinne Antignac and Sophie Saunier. And obviously, he got very excited by genetics, and since his return to his home institute in Budapest, he has been working as a pediatrician, as a clinical genetist, and is currently leading their genetic outpatient clinic, the hemodialysis unit, and a nephrogenetic research group at the Semmelweis University. So Kalman, saying that, the floor is yours, and we are really eager to hear your presentation. Thank you.
Thank you very much, Elena, for this introduction, and let me start on the talk. And dear colleagues, let me please first start with a question. I would like to know who I'm speaking to. So would you please tell me your specialty?
Kalman, can you see it?
No, not yet.
Okay, I will read you the answers. So 60% of our audience are pediatric nephrologists.
60%, 60%.
61%, 36% are adult nephrologists, and we have 3% of others.
Thank you very much. Thank you. Okay. So Mendel was born 200 years ago, and at the age of 61 years, he died of chronic nephritis. So in this year, in this called Mendelian year, I think, especially in this talk, it's worth remembering his famous experiments. So for his experiments, he studied the pea plant characteristics, and luckily he chose binary characters of the pea plant. And when he crossed two pea plants with different characters, what he observed is that in the first generations, the characters are unitary. So they are not bland, as they were if the studied characters were multifactorial, but corresponded to only one of the two parental characters, which he called the dominant character. And when he crossed the first generation, in the second, excitingly, the recessive character reappeared in 25% of the offspring. And this 25% was so nice that many of his opponents did not believe his results, didn't trust his results, because they said that this is too nice, too good to be true. And when he studied several characters, he realized that they reappear in the second generation unlinked, so they were not connected to each other. And based on these findings, he concluded the principles of heredity, which is the characters are determined by hereditary factors, which are nowadays called the genes. And these hereditary factors have alternate forms, which are the alleles. And these alleles are created random and unlinked in the gametes. And so these principles of heredity are still true for monogenic disorders. There are major differences between Mendel's experiments and the heme monogenic disorders. So the pea plant in Mendel experiments must have been an inbred homogeneous population. So it's very likely that a single allele was responsible for each phenotype, and each allele gave 50% of all alleles in each generation. While the human population is highly heterogeneous, so in our population, both the disease-causing and the benign alleles are heterogeneous, allowing many different effects and even interactions between them. And the pathogenic alleles in the human populations are rare, even in autosomal recessive disorders. Nevertheless, monogenic disorders follow indeed a Mendelian transmission pattern. And if the disease-causing allele is dominant and located in an autosome, then the risk of inheriting the disease corresponds to the risk of inheriting the single allele, which is 50%. When two alleles are necessary for the disease development, so the disease-causing allele is recessive, then the risk of inheriting the disease is 50% to the second power, which is 25%. And so this is the typical pedigree in an autosomal recessive disease with affected siblings, at least one of whom is a girl, otherwise it could also be an X-linked recessive disease, and the parents are healthy. So if we further increase the necessary alleles that are required for the disease development, then the risk of inheriting the disease further drops. So in this pedigree, as you can see here, there are 3 alleles necessary for the disease development, also called triallelic inheritance in 2 different genes. In the first gene, there are 2 disease-causing alleles, and then there's a heterozygous allele in the second. And in this case, the risk of inheriting the disease is 50% to the third power, which is 12%. So this shows, if you look at these 3 pedigrees together that we went through, you can see that the higher the number of the alleles required for a disease, the less likely is its inheritance. So the less likely is familial clustering. So it emphasizes why monogenic disorders are clustered in families and why multifactorial disorders, which are determined by several genes and several alleles, are not. On the other hand, you can also say that the more family members are affected by the same disorder, the more likely it is monogenic. So, and this is something very useful in the clinical practice. So whenever we see a child with hematuria and we then know the etiology, then we tend to screen the first-degree relatives, especially the parents, and in case of boys, especially the mother, for hematuria, even if they are unaware of hematuria, because they might be also affected. And if they are, which shows a familial clustering, and it's very likely to have a monogenic origin. And if they are not affected, if the case is sporadic, then it can be multifactorial. It can also be monogenic, of course. While speaking about clustering, familial clustering, and monogenic disorders, I have to add that severe autosomal dominant disorders, despite being monogenic, despite being Mendelian, cannot be clustered in families, because severe autosomal dominant disorders which deteriorate the reproductive fitness of the affected individual cannot be transmitted to the next generation. So they always result from de novo mutations. And as they always result from de novo mutations, a germline mosaicism is rarely cause the affected status of a sibling. Severe autosomal dominant disorders typically appear as sporadic cases, despite being Mendelian. So what are the major differences between the Mendelian and the non-Mendelian disorders? Mendelian disorders determined by a single gene. They are completely penetrant, and as they are completely penetrant, there's a possibility of presymptomatic or prenatal diagnosis, presymptomatic if it's ethical to do. While non-Mendelian disorders, which are determined by several genes and also influenced by environmental factors or stochastic events, are incompletely penetrant, so only a risk might be estimated, which can be also difficult. In monogenic disorders, familial clustering is possible. At least in some families, they do show some typical transmission pattern of clustering, but in the clinical practice, most monogenic disorders appear as sporadic cases. Non-Mendelian disorders in general are sporadic. There's no familial clustering unless the disorder itself is relatively frequent in the general population, like diabetic nephropathy. There's a huge difference in the etiology of end-stage renal disease, clearly explaining the composition of the audience. So I think Mendelian disorders are much more important for the pediatric nephrologists, because around half of the childhood end-stage renal disease cases are due to monogenic disorders. In adulthood, multifactorial disorders as diabetic nephropathy, hypertensive nephropathy, glomerulonephritis cause in the vast majority of the cases the end-stage renal disease. And even the most common Mendelian disorder, ADPKD, is responsible for only 5% of all end-stage renal disease cases in adults. And now the second question.
Okay, the questions are coming. Oh, actually now, yeah, okay. So I will read the questions for you, Kalman, if you don't see them.
Thank you.
So 50% answers between 1 and 5, 21 between 6 and 10, and 23 more than 10.
Okay, so this is not the question. I'm sorry, this is not the
wrong one.
question.
No, but that's the answer to the previous question. How many families have you seen with incomplete genetic nephropathy, or is it?
Okay, the question should have been raised. Do you ask the parents about their potential consanguinity when you consult a child? So would you please raise the third question?
Exactly, I will do. Okay, I'm sorry.
Next up, sorry. Okay, so I will read the question for everybody. Do you ask the parents about their possible consanguinity when you consult a child with a nephropathy of unknown etiology? And then you can select between 4 options. Okay, now these are the right answers. So most of the times, 76%, only if they come from populations with a high degree of consanguinity, 9%. I prefer not, actually nobody. And I'm an adult nephrologist, 15%. Okay, now you can comment.
Thank you very much. Thank you. Okay, so you are absolutely right. I'm happy to hear. Unfortunately, in Hungary, probably because the consanguinity is so rare, many of my colleagues do not raise this question. But this is something I would like to convince all of you to raise this question, especially if you consult a child with an extremely rare disorder, because the risk of having an affected child in consanguineous families really depends on the prevalence of the autosomal recessive disease. So if you think of children with frequent autosomal recessive disorders, they are typically compound heterozygous for 2 variants, unless there's a very frequent variant like in cystic fibrosis. But in other cases, like in PKHD1 associated autosomal recessive polycystic kidney disease, they are typically compound heterozygous for 2 different mutations, because in frequent autosomal recessive disorders, consanguinity, inbreeding is not a major risk factor, but it gets a major risk factor when the prevalence drops, and especially in extremely rare disorders which are not really known even by experts. This is extremely important, and most of the patients who suffer from extremely rare autosomal recessive disease are indeed homozygous for extremely rare variants. So this is an important question, and just remember how much the relative risk compared to the general population depends on the prevalence of the disease. Okay, in the next part, I would like to show you monogenic nephropathies with an unexpected transmission pattern. And before doing that, maybe it's very obvious for most of you, but I remember that at the beginning of my genetic studies, I was often confused with the correct wording. So let me please show you these two pedigrees, which are clearly different. One is X-linked dominant, and there's X-linked recessive. And you can notice that this mutant allele is transmitted in absolutely the same way. So the variant transmission occurs in 50% in X-linked, in autosomal disorders, and in function whether the variant is dominant or recessive, the phenotype is transmitted in a dominant or recessive fashion. So the variant can be dominant or recessive, but this is the phenotype and not the variant that is transmitted in a dominant or recessive fashion. Before going into details, just remember that the transmission pattern depends on the underlying pathophysiology. So if 50% function loss, haploinsufficiency, a dominant negative effect, a gain of function, or a second hit in the Y-type allele is sufficient or necessary for the disease development, then the disease will be transmitted in an autosomal dominant fashion. If the disease development requires a complete or almost complete loss of function, then the disease will be transmitted in an autosomal recessive fashion. But please keep in mind that a protein may be dysfunctional through different mechanisms, and different variants of the same gene may cause a disease through different mechanisms. So this is why the transmission pattern related to the same gene may vary. And this is something we do not really consider as often as it should be. So PKD1 related polycystic kidney disease is very well known to be autosomal dominant disease. In the vast majority of the cases, this is the most frequent monogenic disease. All of you know very well it's dominant. But it has been described, as you probably know, that hypomorphic variants in PKD may not cause a disease phenotype even in the 30s or 40s of the individuals. But when these hypomorphic variants are both transmitted to the offspring, then the children may develop hyperechogenic enlarged kidneys in utero with neonatal respiratory insufficiency, giving the clinical picture of autosomal recessive polycystic kidney disease. So if you look at this transmission pattern, this is clearly autosomal recessive with healthy adults and affected children. On the other hand, MPHS2-associated podocytopathy has been known for a long time to be transmitted in an autosomal recessive fashion. And a few years ago, a family was described with an autosomal dominant FSGS and de novo frameshift mutation MPHS2 that segregated with the disease through 3 generations, clearly showing its causal role. And this is a last exon frameshift mutation, so it's not subject of nonsense-mediated decay. And we found the most frequent variant of MPHS2, R229Q, to be benign and dominant over most pathogenic variants, but recessive with others. So this is the first variant in human genetics that depends in its pathogenicity on the trans-associated mutation. So it's only pathogenic when associated to very specific 3' missense mutations. And in these individuals, the 2 podocin monomers form an abnormal oligomer, which are retained in intracellular compartments, explaining their pathogenicity. So this is a very special aspect in the assessment of the pathogenicity of MPHS2 variants, because here we not only have to know which variants are pathogenic, which variants would be pathogenic in the homozygous state or when associated to other loss of function mutation, but we also have to assess which variants among the pathogenic variants would also be pathogenic with R229Q. And as we know that it occurs through an altered oligomerization, we consider a variant to be damaging, to be pathogenic with R229Q if it affects an evolutionarily conserved amino acid, It's located in the region of oligomerization, so in the C-terminal region of podocin. The substitution changes the biochemical properties of the C-terminal helical regions without disrupting the oligomerization. The oligomerization, despite being the cause of this dominant negative effect, is needed for exerting this dominant negative effect. So the mutations cannot disrupt the oligomerization. Okay, and here the second question should have been raised, but as far as I understood, many colleagues in the audience have already seen several families with complete penetrance.
Should we ask it quickly again?
No. Just tell me please the answers because I missed them.
54% said between 1 and 5, 21% between 6 and 10, and 23% more than 10.
Okay, so I do agree that incomplete penetrance is rare in nephropathies. So there are only a few examples for that. So one of the most exciting examples for incomplete penetrance in nephropathies is the PIC1 associated nephrotic syndrome, which has been described incompletely penetrant by 3 different research groups. And what is fascinating in this nephrotic syndrome is the unaffected individuals carry homozygous loss of function mutations. So these are not only hypomorphic variants which allow some residual function, but these are truly loss of function mutations with probably completely abolishing the protein's function. And despite of this, several families, 6 families altogether, have been described to have unaffected individuals. And nobody knows, as far as I know, the explanation for this incomplete penetrance. Another example for potential oligogenism was described in Alport syndrome. So this was done by Italian and several European groups. And 11 families were described in this very nice paper with digenic mutations in more than 2 Alport genes. And also what they observed is that the phenotype in individuals with mutations in more than 1 Alport genes, in 2 Alport genes, is more severe than those who are heterozygous for a single mutation, a single gene. And this is very nicely explained by the fact that patients with digenic mutations, digenic heterozygous mutations, have 25% normally formed collagen trimers, while those who have heterozygous mutations have 50% normally formed trimers, and those who suffer from an autosomal recessive form of Alport syndrome have no functional, have no properly folded trimer at all. Another example for digenic inheritance is provided by Bartter syndrome. A single child was described in the original description who had mutations once again, similarly to COL4A3 and COL4A4 in 2 neighboring genes. But in this time both alleles were mutated in both genes. So mutations of the first gene encoding a chloride channel would cause a MyD beta insipidus on its own, and mutations of the second would cause a classic Bartter syndrome type 3. But when both are mutated because of 2 point mutations on the parental allele and the common deletion affecting both genes on the maternal allele, not only a more severe phenotype develops, but also deafness appears, probably because the 2 chloride channel encoding genes would complement the loss of the other when only one of the 2 is mutated. But when both are mutated, they cannot compensate each other anymore, and so the inner ear gets also affected. MPHP1 is by far the major gene of nephronophthisis, and its biallelic mutations cause isolated nephronophthisis in more than 90% of the cases. But rarely neurological symptoms of Joubert syndrome or retinitis pigmentosa are associated. And well, we and others have suggested the role of second locus modifiers, and we screened several genes for heterozygous variants, and we found in a Joubert syndrome gene AHI1 heterozygous missense mutation in patients who also suffered from neurological symptoms. These are small cohorts, but the same result was found by American colleagues in patients with MPHS1 mutations and retinitis pigmentosa. They also carried in a much higher degree this R830W variant than in patients isolated nephronophthisis. And they also generated a nice mice model, and they showed that the mice is also knockout for MPHP1 and also carries a heterozygous AHI1 mutation, develops a more severe retinal involvement than the knockout, than the MPHP1 knockout, indicating the modifier effect of AHI1. But probably the most fascinating example for oligogenesis was provided by the triallelic inheritance pattern in Bardet-Biedl syndrome. And in this original description, the most convincing family was this one, with two brothers who were both component heterozygous for a nonsense mutation, but one of the two was unaffected, and this unaffected brother did not carry a second locus mutation that was carried by the affected brother. So the affected brother had 3 nonsense mutations, while the unaffected was only component heterozygous for 2 variants. So this is indeed attractive to explain the unexplained status of this brother by the lack of this 3rd mutation. But this triallelic inheritance was reassessed by several groups, and it hasn't really confirmed the existence of this triallelic inheritance in Bardet-Biedl syndrome. And so when it was studied in large patient groups and compared ciliopathy and control groups, in this first one, additional variants were looked for in second ciliopathy genes, which were rare, or not only rare, but also predicted to be damaging. And a very similar mutational load was found in the ciliopathy and the control group. So when it was properly done, there was no difference between the ciliopathy and the control group, which would show the effect of a 3rd heterozygous mutation. And in another study, which used different approaches, and I only show one of them, when single heterozygous rare deleterious variants were looked for in at least 2 different Joubert syndrome genes, so corresponding to a degenic inheritance, a very similar mutational load was found in patients with Joubert syndrome and in the control group. So there's no difference in the mutational load between patients with ciliopathy and healthy individuals. So this probably oligogenism is not as important in ciliopathy, as previously suggested, and ciliopathies are at least in the majority of the cases, are Mendelian disorders. So what were the reasons that explain the oligogenesis theories in ciliopathies? There's indeed a wide variation in expressivity in the ciliopathies. So second locus modifiers do exist, probably exist. Well, probably the most important point is that during the first decade of this century, it was really challenging to assess the pathogenicity of rare variants, simply because we did not have this large population databases of the variants. We couldn't really assess the frequency of rare variants. We screened a few hundred controls, but we didn't have thousands of controls as we do have nowadays. So many of these rare variants were simply falsely considered pathogenic. And finally, ciliopathies are genetically highly heterogeneous, and so the higher the number of the causal genes in a disease, the more frequent are heterozygous carriers in the general population. So this graph shows this relationship between the number of the causal genes and the proportion of the heterozygous individuals in the general population. As you can see here, a disease with a prevalence of 1 to 50,000, if it had a single causal gene, the heterozygous carriers in the general population would be around 1%. But with such a high number of genes as nephronophthisis and Bardet-Biedl syndrome have, the heterozygous carriers are in the rate of several persons. Maybe this is somewhat overestimated, because when I calculated this, I assumed that the genes are mutated with a similar frequency, which is not true. But I also assume that all variants are completely penetrant, which is not true either. So one of the effects makes me overestimate, the other underestimate the rate of the heterozygous carriers in the general population. But the major message is that in the general population, several percent of the general population is a heterozygous carrier of a true pathogenic mutation of nephronophthisis or Bardet-Biedl syndrome or ciliopathies, which are genetically highly heterogeneous. So even if you find a mutation in a second locus, in a second gene, it doesn't necessarily mean that it is causal. It doesn't necessarily mean that it contributes to the phenotype. It may be accidental, because even in the general population, we can find causal variants by chance. And this is why it was relevant to compare the control groups and the ciliopathy groups this way, also comparing the predicted to be damaging variants in the two populations. We saw how much the transmission pattern can depend on the causal variant and the physiology and the pathophysiology. Nevertheless, most of the autosomal recessive disorders are considered to be completely penetrant, and very few variants were described to be incompletely penetrant in autosomal recessive disorders. So we raised this question whether in autosomal recessive disorders there can be other incompletely penetrant variants. And we developed a population genetic approach that I would like to show you here. So basically, we compare the relative allele frequencies of the pathogenic variants in the patient population and in the general population. But here you can only see the pathogenic alleles within a gene. So this is why they are so comparable. So you can see the most well-known mutation of CFTR responsible for cystic fibrosis is the F508 deletion, which is responsible for more than half of the mutant alleles. So its relative frequency is similar in the general population, among the pathogenic mutations, and in the patient population. And the loss of function mutations have a similar contribution in both populations. But if you look at this well-known incompletely penetrant variant in CFTR, the R117H, you can see that it's relatively frequent in the general population. It's as frequent, almost as frequent as the other LOF mutations, as the other loss of function as the loss of function mutations in the general population. But in the patient populations, it's 5 times less frequent. So by comparing the relative contribution of each variant to that of the loss of function variants in the patient population and the general population, according to this equation, we can calculate the penetrance of the variants, or we can at least estimate the penetrance of the variants. And as you can see here, for this variant, we calculated a penetrance of 21%. So we analyzed this using this method, the relative allele frequency of 2,000 variants in 17 autosomal recessive disorders. And on this graph, you can see that the variants that are located under first I would show you the completely penetrant variants. So the completely penetrant variants are equally represented in the general population and in the patient population. So this is why they are located along this line. The variants that are underrepresented in the patient population versus the general population are located under this line. So these are the incompletely penetrant variants, which are not as frequent in the patient population as they are expected to be based on the general population allele frequency. So we studied 3 nephropathies among these 17 autosomal recessive disorders. We didn't find any incompletely penetrant variant in MPHS1 associated nephrotic syndrome. We found only a single one in MPHS2, which is RT2-NIQ. It doesn't mean that there are no incompletely penetrant variants in these 2 genes, because this population genetic approach is only informative for the frequent variants. So the rare variants can easily go undetected despite being incompletely penetrant. So this is a major disadvantage of this approach currently, and this is why larger and larger patient cohorts will be necessary to analyze to identify further incompletely penetrant variants. But with PKG1, with a large number of patients in the medical literature, we could identify 4 missense mutations that are incompletely penetrant. As you can see here, this column shows their enrichment in the patient population versus the general population. So you can see here that these variants are more frequent in the patient population than in the general population. Absolute frequency these are. So indicating that these are indeed pathogenic variants. So these are not falsely published benign variants. But despite being pathogenic, they are much less frequent than expected based on the population allele frequency, giving a penetrance between 3 and 17%. And among these variants, you can also find the I2331K variant of PKHD1, which is quite a well-known mutation, known to be hypomorphic. All these are hypomorphic mutations from a clinical point of view, and in accordance with their incomplete penetrance. So it has a penetrance of only 16%, which looks clinically relevant to know. So I think as we have more and more information about the effect of specific variants, though we currently say that MPHS2 related nephrotic syndrome is autosomal recessive, and this is true for the vast majority of the variants, we have to keep in mind that the transmission pattern depends on the variant. So in some cases, it can be an autosomal dominant FSGS. So this is why the proper description of the transmission, not only the gene, but also the phenotype and the variant are needed. Further examples, as you saw, the PKD1 non-mutation related polycystic kidney disease is indeed autosomal dominant, but this hypomorphic variant related polycystic kidney disease, as you saw, was autosomal recessive. The PKHD1 T36M, the most frequent variant of PKHD1, is a completely penetrant autosomal recessive disease, but the PKHD1 I2331K related polycystic kidney disease seems to be incompletely penetrant. The MPHP1 homozygous deletion-related nephronophthisis is indeed autosomal recessive, but the same mutation-related retinitis pigmentosa is multifactorial, depends on second-look mutations which do not even explain the whole clinical picture. So other factors also play a role in the development of retinitis pigmentosa. So this is why it's important not only to define the gene, also the mutation, and also the phenotype that we speak about. And finally, let me show you an example for a multifactorial nephropathy. Now, it was known for a long time that end-stage renal disease is more frequent in the African American population than in the others. And nobody knew the explanation. And around a decade ago, it was very nicely found by GWAS studies that 2 alleles, the G1 and the G2 alleles of APOL1, provide this risk. These are responsible for this increased risk. These 2 alleles, G1 means 2 missense mutations, 2 missense variants, and G2 is an inframe deletion. And these alleles are much more frequent in the African American general population than in the European or Asian or Latino populations. And they are especially frequent in African American individuals who are affected by FSGS. And why is it so frequent in the African American population? And the very nice explanation was given for this high prevalence, is that in the heterozygous state, these 2 alleles, G1 and G2 alleles, provide a protection against the Trypanosoma brucei rhodesiense related sleeping sickness disease, sleeping sickness. Unfortunately, even in the homozygous state, they cannot protect against the Trypanosoma brucei gambiense. But in the homozygous or the compound heterozygous state, so if both alleles are carrier, and both alleles, either G1 or G2 alleles are found, then there's an increased risk of hypertensive nephropathy, FSGS, or HIV-associated nephropathy. And the lifetime risk in these individuals is 4%. So this 4% clearly indicates the difference between a Mendelian disorder and a multifactorial disorder. So a Mendelian disease with complete penetrance, individuals who carry pathogenic alleles and both alleles, pathogenic variants and both alleles, they have 100% risk, lifetime risk to develop disease, autosomal recessive disease, where it's only 4%. 4% in multifactorial disease is very high, but it's clearly the difference between a Mendelian and the multifactorial disease. And if an HIV viral infection is associated, then the risk is elevated to 50% if it remains untreated. So in conclusions, nephropathies are typically either Mendelian or multifactorial. Oligogenic inheritance is rare, but variants at second loci may modify the phenotype. It seems worth describing the transmission pattern in function of the causal variant and the organ involvement, and not just in function of the causal gene. And incompletely penetrant variants can be detected by a population genetic approach in autosomal recessive disorders, and are more frequent than expected. So probably with the further studies of more autosomal recessive diseases and in large patient cohorts, we will be able to identify more incompletely penetrant variants, which are crucial to know in the genetic counseling. Thank you very much for your attention.
Lena, you are muted.
I'm sorry. Yeah, so thank you very much for this wonderful presentation. I think that our life becomes even more complicated with knowing about these incomplete penetrant variants that we are probably even unable to classify the disorders, as previously we thought to have a clear-cut autosomal dominant, autosomal recessive, X-linked, but now it becomes a little bit mixed. So, but I think your approach is really good and I think this approach can be used because I think all of us who have now access to whole exome sequencing or large gene panels, we have to address these variants of unknown significance, and there are a lot of them in many genes. So do you think that this approach you propose to compare the allele frequency of these variants of unknown significance in populations with disease and without disease, can it solve that problem, or it's only restricted to some specific variants with incomplete penetrance?
It allows us to reassess the pathogenicity of the variants in autosomal recessive diseases. So we do use it to reassess, first of all, to confirm the pathogenicity, to show that these are more frequent in the patient population than in the general population. So we did exclude many variants that were published and are still published in the Human Gene Mutation Database that are actually not enriched in the patient population. So we excluded these variants. And this approach, I think it's indeed useful, and we work on generating an algorithm that's accessible for everyone because what it needs is a large population, large patient populations. So for this analysis, we collected 12,000 patients, genotype data of 12,000 patients mutated in one of these 17 or affected by one of these 17 autosomal recessive disorders. But whenever somebody wants to study and has a huge patient cohort, we would like to allow them to upload or just to use this this algorithm and to compare, because well the ratio in itself that I showed you is quite simple, but we used many corrections to correct the degree of consanguinity to exclude the non-pathogenic variants. We analyzed whether there's a possibility of incompletely penetrant loss of function mutations, because we compare the allele frequency to the loss of function mutations. So if the loss of function mutations are incompletely penetrant or lead to in utero death, this is no longer usable, this approach. So these are the points we have to verify before using this population genetic approach.
Mm-hmm. Okay, thank you. Well, other people, I think the question was asked in the beginning of the presentation. I think you answered it more or less, but I will just read the question, so maybe you can comment. Thank you for your excellent presentation. How much Mendelian inheritance would explain variability in phenotypic expressivity of a particular condition? So you talked a little bit about it. Can you follow the question, Kalman?
No, will you please repeat it? Sorry.
So how much Mendelian inheritance would explain the variability in the phenotype in a certain condition? So I think it goes about the severity of mutation and their Mendelian inheritance. So if you have a condition in the family with a certain mutation having a very different phenotype from light to severe, to which extent the Mendelian inheritance can explain that? I think that is a question that.
So I think well, the majority of the variability in the phenotype is in autosomal recessive disorders, but even in autosomal dominant. So in Mendelian disorders, anyway, is determined by the type of the variant. So and this
yes,
is
I think in the same condition, okay, probably if you inherit the same variants, then the variants that there may be some modifiers that can modify your phenotype. Right?
Yes, this is something that can be answered by family studies. So if you compare, whenever you see a difference in the phenotype between siblings or between family members carrying the same mutation, it must be the effect of second loci variants and not by the primary gene itself.
Yeah. Another question is about de novo variants. So the question is, many of the variants reported in monogenetic kidney diseases are novel or de novo, and what is the risk of false identification of these variants as being pathogenic? So we come back to your to the discussion.
Okay, so this is something. What for the analysis for the judgment of these de novo variants, what has to know is that each of us inherit 40 to 80 de novo variants, depending on the paternal age. So out of which 1.5% is located in an exon. So in brief, around 1 to 2 mutations are located, de novo mutations affect or 20,000 genes. So whenever you find a de novo mutation in a gene that is compatible with the phenotype, this is very likely to be causal, because the risk of having a de novo exonic mutation in a gene that corresponds to the phenotype is extremely low. So this is why the de novo appearance is such a strong argument for the pathogenicity, supporting the pathogenicity of the identified variant.
Okay, I think it's clear. So just scrolling through other questions. Yeah, I don't see more questions. So another question I had: how important to have to study the population of origin? For example, if you have identified, even if you are applying your approach to compare complete and less complete penetrant variants, how important is to have, for example, if it's described in the African population, to have a control population, the African, because I think this is a big issue also. We don't have a lot of data in certain populations, making it difficult to interpret genetic data.
Absolutely. So for the population genetic approach, it's very important to compare the same population. So in our approach, I didn't specify, but we only compared the European patient populations and the European general population. But even within Europe, there are differences. We know very well that there are some variants which are frequent in Western Europe and others in Eastern Europe. So, but currently, as we could only assess the penetrance of the frequent variants, this is not such a huge bias, because the frequent variants appeared very early in the colonization of Europe. So they are quite well distributed. So for the assessment of the frequent variants, this is not a huge problem. But the less frequent variants have to be compared with the proper control population. So absolutely, that's very important. And this is why we need to have not only large population reference populations, but also more and more patients in the patient population to properly estimate the penetrance. This is the major disadvantage of the population genetic approach. Your question is absolutely right. Thank you.
Okay, thank you. And just last question, maybe, because a lot of our of people in the audience are clinicians. So can you provide some guidance, just in more general terms, for the clinicians? If you have a patient with a certain genetic condition, would you still perform their Sanger sequencing of the gene, or if you, for example, have a patient with, I don't know, clear disorder that like a single gene mutation, or you still would recommend a gene panel or whole exome sequencing, because then you can discover more pathogenic alleles. Huh?
Well, if the phenotype is really suggestive of directly of a gene, and this is still the case in many families, or there's a very common cause of nephroptosis. So this screening WT1 mutations in patients with the clinical diagnosis of Dennis-Dress syndrome, Frazee syndrome, this is quite obvious, and there's no need for panel sequencing. Or if you see a child with clinical diagnosis of nephroptosis, we did not do a panel test, but we just screened for the MPHP1 deletion, which is by far the most common mutation in end-stage renal disease in childhood. So, but otherwise, if at least 2 or 3 genes can be implicated in this disorder, or we do not find the most frequent mutations in the major genes of the disease, then we skip to panel sequencing or even exome sequencing.
Okay. Thank you so much. I think several people thank you for the excellent presentation. So I also thank you, and I invite everybody to attend our next webinars. And thanks a lot. We learned a lot, and yeah, I wish you all a very good evening. Bye bye.
Thank you very much. Thank you. Bye.
Thank you. Bye.